Thank you to the organizing committee for giving me the opportunity to present my work today. My name is Jantarika RGJ. I am a PhD candidate at Biochemistry Department, Faculty of Science, Mahidol University. Today I would like to talk about my first publication in the title of Single-Cell Temporal Analysis of Natural Dengue Infections, Review Skin-Homing Lymphocyte Expansion 1 day before differescence. Dengue infection is now a significant problem and present in several tropical and subtropical countries, including Thailand. Half of the population in the world are considered at high risk of dengue fever, as you can see in the red color here. The clinical phase of dengue infections can be divided into three phases. The first phase is febrile phase. At this phase, patients will have high fever and concentration of virus is high. Immune cells such as T cell and B cell are activated and help to eliminate the virus out of our body. The second phase is critical phase when the body temperature of patient return to normal. At this point, the concentration of virus is almost gone. However, the immune cells are still activated and these phenomena can promote internal bleeding, give a high risk of shock and potentially death. The last phase is recovery phase when patients return to normal. Unfortunately, there is currently no specific treatments or established molecular marker that can be used to predict the disease progression. Understanding biomolecular changes of the high complexity and heterogeneity in immune cells over the time course of dengue infections is necessary for effectively clinical management and may help dengue patients. So to fill this gap of knowledge, we have applied the single-cell RNA sequencing technology to investigate the transcriptomic changes of immune cells across multiple time points during dengue infections. Why we perform the single-cell RNA sequencing is because let's say we have high heterogeneity of samples and then we pull them together. So that means we're going to get average gene expression from all cells. And moreover, we are not able to identify the cell types as shown here. However, if we perform single-cell RNA sequencing, so that means we capture individual cells. We can explore the expression of individual cells and also we can group them and identify cell types based on whole expression. First of all, I would like to thank Dengue Thailand that provides us a very well-defined cohort. We obtained the blood sample from two dengue patients, one with dengue fever or DF, the other with dengue hemorrhagic fever or DHF. The samples were collected at 4 different time points, including differescence or 1 long, 1 day and 2 days before differescence and 2 weeks after differescence. In this study, we also include 2 healthy donors as the control. After we collected the sample, we then construct the single-cell RNA library using a doublet-based platform from 10x company. Raw reads were then sequenced and mapped into human reference genomes, and a computational analysis was performed. We identified the immune cell type based on their whole expression profile as shown here. This spot represents the similarity or differences between whole expression profile of individual cells. One dot represents one cell, X and Y axis represent the distance between individual cells. From here, we could identify T cell, red blood cell, plasma blast, B cells, NK cell here, monocytes, and dendritic cells. We next explore the proportions of each cell across the time course of dengue infection. Y-axis represents the percentage of cells, X-axis represents the date of infections. Here represent DF, DHF, and healthy controls. Different colors represent different cell types. T cell expanded the mode at the clinical phase or DEF in both DH and DFH, whereas plasma cell and plasma blast expand the mode at day -1 or 1 day before fever subsides when compared to recovery phase and healthy control. Next, we ask how this population dynamic reflects the biological functions across time course of dengue infections. We further look at the dynamics of gene modules that are associated with biological pathway across time of infections. Y-axis represents the significant biological pathway, whereas X-axis represents date of infections. Here represent DF, DHF, and healthy control. Different columns represent different cell types, monocytes, NK cell, T cell, plasma cell, and plasma blast. Genes that are associated to type 1 interferons pathway are regulated at DMNAT2 and decrease toward differescence in all cell types, but this pattern is more prominent in monocytes. Apart from the pathway that share in all cell types, we also observe the cell type-specific pathway. We found the upregulations of monocyte-specific pathway peak at DMNAT2, including cytokine productions and inflammatory response. At DMNAT1, we found peaks of genes that are specifically expressed in plasma cell, plasmablasts, and B cells, including B cell-mediated immunities and humoral immune response. However, from this analysis, we didn't see any specific pathway to T cells. It might be because of high heterogeneity of T cell populations. So we next further analyzed early T cells and we were able to identify subpopulation of T cell as shown here. There are two angles of cells shown here. These angles are the group of naive-like T cell, including naive CD4 and naive CD8, whereas this angle presents the group of effector-like T cell, including effector CD4 and effector CD8 group 1, 2, and 3. We look at the differential gene expressions across T cell population as shown here. We found that effector CD4 and effector CD8 group 2 share similar expression modules. Interestingly, these two subsets of effector T cells show their tissue homing signatures by upregulation of adhesion molecules, including TUBB and TUBA1B genes. We speculated that effector CD4 and effector CD8 group 2 might express the tissue homing genes that are associated with the potential sites that they might home to. We further explored the expressions of marker homing genes at different tissue and found that during the acute infection, several tissue homing genes are upregulated. Interestingly, at DMNAT1, skin homing gene, which is SELPLG genes, is upregulated in these two effector subsets of T cells, suggesting that they might be able to home to skin, the initial sites when virus infect in our body. We may question that sometimes transcription is not always correlated with translation level, so we further validate these skin homing genes in protein level. We perform flow cytometry to detect the expressions of CLA molecules or skin homing molecules at the surface of the cells. From this, we observe significant expansions of CLA positive, CD69, PD1 positive in both CD8 and CD4 T cells at DMNAT1 when compared to weak follow-up and healthy. Our study provides a comprehensive understanding of dynamic immune responses in natural dengue infections. Genes that are associated to innate immune response are upregulated at DMNAT2, whereas genes that are associated to adaptive immune response peak at DMNAT1. Moreover, we show that subset of effector T cell upregulated skin homing molecules in both transcriptions and translation level, suggesting that they might home to skin, the initial site, at 1 day before critical phase. Last but not least, this research would not have been possible without all the support. I would like to thank Dr. Walodom Chalansawan, Dr. Ponpan, Dr. Nanya, Dr. Sarah, and all the lab members. I would like also thank all the fundings, especially RGJ PhD scholarships, and thank you for everyone for attending my presentation. I would happy to answer any questions. Thank you.